Okay, so welcome back from the break. Let's take a look at the concept check questions, and then we'll look at something else that came out of this particular piece of work that Morgan was doing. So which of Mendel's laws describes the inheritance of alleles for a single character? Well, that would be the law of segregation, or law of separation, if you want to use that term, but we usually say segregation. And which law relates to the inheritance of alleles for two characters? Well, that would be the law of independent assortment. What is the physical basis for each of Mendel's laws? Well, the law of segregation, remember, is based on the behavior of chromosomes, the homologous pairs of chromosomes, in anaphase 1, where the homologous chromosomes are separated from each other. So the physical basis for that would be the separation of the homologous pairs in meiosis 1, specifically metaphase 1. What would be the physical basis for the law of segregation? Well, that would be the independent and random arrangement of the chromosomes on the metaphase plate law of segregation is anaphase 1, isn't it? I misspoke on that one. So the law of segregation is the separation of the homologous chromosomes in anaphase 1. So maybe I should write this in. And law independent assortment. That, of course, is metaphase 1. And remember, again, that's where the chromosomes line up independently and randomly on the metaphase plate. And that's for two characters. I need to make sure I stop and think before I say things, I guess. So, law of segregation explains the behavior of single characters. Law of independent assortment explains the behaviors of two characters. The physical manifestation of the law of segregation is the separation of the homologous pairs of chromosomes in anaphase 1. And the physical manifestation of the law of independent assortment is the independent and random arrangement or aligning of the chromosomes on the metaphase plate in metaphase 1. So it's just a reminder of the basis for Mendel's laws, because we're going to see those at work today still. Or we're going to be looking at some variations from that. So what we have now from Morgan's work and from Hetty Jake's work and other people's work is that there is a chromosomal basis to sex or gender. And in insects, or in some insects, and in mammals in general, it's the X and the Y chromosomes. So for insects, the X and the Y are distinctly different from each other in terms of their shape. In mammals, and this includes humans, the X is quite large and the Y is quite small. And so there's a whole region of the X chromosome So there's this whole region of the X chromosome that has no matching region in the Y chromosome. So whatever genes and alleles of those genes that happen to be present in the X chromosome are the alleles that the male is going to show. And when we look at the insects, it's the same idea. There's a region in here on the Y chromosome that has no matching gene region for what's on the X chromosome. And so any genes that happen to be in this region of the X chromosome and the alleles of those genes are what the insect males are going to show. And that's where the eye color gene that gives the white eye mutation happens to be located. But the XY system is not the only system of gender determination. So here's our system, the XY system. In many insects, it's what's referred to as the X0 system, where there are two X's in the female and only one X in the male. So again, if you have a true homologous pair, you are a female. If you have a single chromosome, then you're male. In birds, and here we're looking at chickens, it's the female that has the odd pair of gender chromosomes and the male that has the two matching homologous gender chromosomes. So it's flipped around in the other way. So the females are the ones with the odd pair of gender chromosomes, and males have the true homologous pair. In communal insects or colonial insects, like bees, wasps, termites, and ants, the females are all diploid, and they're the product of fertilized eggs. The males are all haploid, and they're the product of unfertilized eggs. And this is the reason why when Mendel turned to trying to breed honeybees, he was unsuccessful, because he did not realize that there was this difference in terms of how you got male versus female communal insects. The females are all diploid, the males are all haploid. And so all the techniques that he had learnt, all the principles that he had learnt from observing his garden peas simply wouldn't work on honeybees. And so he was not successful at breeding honeybees and improving honeybees. All his breeding experiments failed, as best we can tell, as far as honeybees are concerned. But these are the other sex determining, or some other sex determining or gender determining chromosomal systems that are used in other organisms. But we spend most of our time talking about the XY system because this is ours. So let's take a look at what happens to traits that are associated with the sex chromosomes. What inheritance patterns do they actually show? Now, it turns out that there are about 10 genes on the Y chromosome. One of those genes, known as the SRY gene, is the one that is responsible for determining maleness. It turns on at around week 6 of pregnancy, and it stays functional for about 3 days. And during that time period, it establishes the cascade of events that will lead to the development of male sex characteristics. So the development of the penis, testicles, all of the external features that we associate with maleness. That's triggered by the function of the SRY gene. Now, that does not always work. If the SRY gene does not function, or if for some reason there's insufficient testosterone produced, or it turns out that the testosterone has no effect on the cells, then the fetus defaults to the default setting of development, which is female. And while you may find this rather unusual, this is in fact something that we know occurs because there are a significant number of females in the population who have a Y chromosome. They are chromosomally XY. But the function of the Y chromosome, and specifically the SRY gene or the testosterone or the ability of the cells to react to testosterone failed to occur, and so they followed the female developmental pattern, which is the default pattern. There's a lot of interesting things we could talk about here, but I don't want to get us too tied up in this for now, because we have a lot more to discuss in this chapter. But we do know that there are other genes on the Y chromosome other than the SRY gene, but the SRY gene is the one that is central to the development of maleness. On the X chromosome, there are a couple of genes that are associated with femaleness, but they are overridden in the presence of the Y chromosome. But in the absence of a functional Y chromosome, for one of the reasons that I've given, that X chromosome then becomes the one that determines the overall fate of the embryo, and it develops as a female. But again, there are other genes on the X chromosome, and there's that whole region of the X chromosome that contains genes that determine traits that are going to be seen in the embryo and seen in the individual that develops from that embryo. And because in the male, there are only those genes on the X chromosome, and it's the alleles of those genes that are going to be expressed, well, that means that's what the male individual is going to show. And so that is going to change how we have to look at the inheritance patterns. So in this diagram, we're trying to show how the inheritance patterns play out under different circumstances, depending on where the X chromosome is coming from and the nature of the X chromosome, because it's really all about the X chromosome. So here we have a female who is normal. Notice that she's X big N, X big N. Actually, I'm going to pull this down. I don't know whether you can see that box, but I can. I'm going to put it at the bottom for now. So you have a female who is X big N, X big N. So she is wild type. She's normal, and she's homozygous normal. And here's a male who has the condition. So he is X little ny. And remember, he only gets one copy of the X chromosome, and it's whatever allele is on that X chromosome that determines his outcome. So if he has the mutation, he's going to have the condition. So this leads to an important observation that we need to make here right now, before we go any further. So while females can be carriers of X-linked traits, males can never be carriers. And the reason for this is quite simple. Remember, the females have two X chromosomes. Females always have two X chromosomes. So they could be heterozygous and still have the normal trait, but be carriers of the allele for the recessive trait, which is by definition what we mean by carriers. Males, on the other hand, can either have the normal trait and the allele for it, or have the allele for the recessive trait. That's the only alternatives that they have, because remember, they are monozygous. They only have one copy of the allele. Yeah. That's what we mean by monozygous. So they can't, a male can never be heterozygous for one of these genes. They can only be monozygous, have a single copy of these genes. either of these alleles, either the allele for the wild type trait or the allele for the mutation. And whatever allele they have, that's the allele that they're going to express, and that's the trait they're going to express. So this male has the X little n, he's got the trait. He passes his X on to all of his daughters. His daughters also receive an X big N from their mother. So they all turn out to be X big N, X little n. So they are all carriers. The daughters are all carriers. The males get the Y from their father and get their X from their mother, and they get an X big N because that's all she has. So they are normal. They don't have the trait, they're normal. The females, however, are all carriers for the trait. So here's the next situation. And now I'm going to move this box back up to the top again. A carrier female with a normal male. So again, the males get a Y from their father. They get their X from their mother. And because their mother is a carrier of the trait, half of the males get the X big N, half of the males get the X little n. So half of the males show the trait, and half of the males are normal. The daughters all get the X big N from their father, and then from their mother, they'll either get an X big N and be homozygous normal, or get the X little n and be heterozygous, showing the normal trait but carrying the allele for the recessive trait. So they're all carriers. So it's a 50-50 split as far as the females are concerned, 50% homozygous normal and 50% showing the normal trait but being carriers, heterozygous. For the males, it's also a 50-50 split, 50% normal and 50% with the trait. Notice no carriers, because the males can never be carriers in this situation. Here's the circumstance under which you can get a female with the trait. But it's a very specific situation. The mother has to be a carrier of the trait and the father has to have the trait. Because remember, for a female to have the trait, she's got to be homozygous for it. And here is the homozygous daughter. Who has the trait. She got the X little n from her mother, and she got the X little n from her father. That's the only way that you can have a female with this trait. And a number of years back, I guess it would be getting on for about 8 to 10 years ago now, I had a student in my class, female student in my class, who was red-green colorblind. And when we started talking about this situation, she was quite excited because she finally understood why she had red-green color blindness. It was because her mother's father, in other words, her maternal grandfather, was himself colorblind, which meant that her mother was a carrier of the allele for red-green color blindness, and her father, my student's father, was himself red-green colorblind. So she got the colorblind allele from her father via his X chromosome, which is what he must pass on to her. And she also got the colorblind allele from her mother, because her mother had a 50-50 chance of giving it to her. And this is where my student sat on the Punnett square. X little n, X little n. She was very excited because she finally understood why she was red-green colorblind. I never asked her whether she had any brothers who were red-green colorblind, but the chances were 50-50 if she did have brothers that they would also be colorblind, because you can see here, half the male offspring are normal, half the male offspring are colorblind. And then the other 50% of the female offspring are carriers, because they get the X big N from their mother, they get the X little n from their father. So the source of the mutation, as far as the males are concerned, is always the mother, because they get their X chromosome from their mother. So the source of the mutation is also always via their mother, because if they can't get the X from their father, they must get the Y from their father, because if they didn't get the Y from their father, then he would be a she, because they would get the X and from their father, and they would get the X from their mother, and that would make them female, not male. So they must get the Y from their father in order to be male. So it's all about the X that they get from their mother. And that means when we start looking at the traits that follow these patterns in humans, there is reason for some concern. Red-green color blindness, and we need to be very specific here, because there are a number of different forms of color blindness, but it's red-green color blindness that is specifically associated with the X chromosome. Red-green color blindness you can live with. I actually used to work for a man who was red-green colorblind. And he explained one day how he got around the limitations. He simply memorized where the red and the green lights were on the traffic lights. If they're vertical, red is at the top, green is at the bottom. If they're horizontal, red is to the left, green is to the right. And he just simply looked at which was the most intense in terms of brightness. That was the one he knew was on, because he saw them as being grey. And so all he had to do was look at which one was the most intense, and that determined whether he would stop or go. And he also had his wife lay out his clothes for him so that he made sure that he didn't put the wrong colours together. Otherwise, he just lived a perfectly normal life, and most people with red-green colour blindness can. There are very few limitations on people with red-green colour blindness. One of the major ones is that you can't be a pilot because you can't distinguish the red and green marker lights that they use to identify left and right. But there are very few other limitations as far as people with red-green colour blindness are concerned. Haemophilia is a little bit more of a concern, because in haemophilia, your blood doesn't clot. And there are two different forms. And both are genetic. And they affect clotting factors. Oops. I won't write them in Roman numerals. I'm going to do them in Roman numerals, which we normally do. I'm not going to do them in Roman numerals because you may not recognise them. So it's blood clotting factors 8 and 9 are the ones that are affected. This was at one time known as the Royals disease, because it was found amongst the royal families of Europe. We think it originated with Queen Victoria and was passed on into the royal families of Europe via her daughters, who married into some of the major royal families, including the Russian royal family. And that's where it has a significance in world history, because the young son of the last Tsar of Russia was a haemophiliac, and his parents were extremely worried about his ability to assume the throne when his father died, and they spent a lot of time fussing over the boy and protecting him, and basically ignoring the country. Not as if they paid a whole lot of attention, but they really did ignore the running of the country and paid attention to their young son, which would eventually lead to the Russian revolutions of 1918 that brought the communists to power. Duchenne's muscular dystrophy is the really bad one, because this is a neuromuscular condition. It leads to loss of muscle control. It starts at the extremities, so it starts at the feet and works its way up. By the time these young males are around 8 to 10, they're probably paralyzed from the waist down and sitting in wheelchairs. By the time they get to 18 or 19, they're paralyzed from the neck down, and they're dead by the time they're around age 22 or 23. And it must be totally devastating to the mothers to realize that the allele for this particular condition came from them via their X chromosome, because that is the nature of the inheritance pattern here. It follows exactly the same inheritance pattern as color blindness and hemophilia. It's on the X chromosome. This is the one which probably would be very unusual to find in a female, by virtue of the fact, remember, that the only way to have a female with the condition is for the mother to be a carrier of the condition and the father to have the condition. And by the time a male with Duchenne's muscular dystrophy was old enough to consider fathering a child, he would already be partially, if not completely, paralyzed. And I'm not sure that any couple would particularly want to have children as a consequence of this anyway, because it's basically saying you're going to have children, 50% of your children, half your children will potentially have this condition, and it is fatal. Not a particularly pleasant thought. But this is the nature of these diseases. And again, remember, the individuals who are predominantly affected are the males, and this is because they get their X chromosome from their mother. They get their Y chromosome from their father. So this is a condition that is passed on from mother to male child, which obviously makes it quite devastating to the parents, particularly to the mother. Now, there's one other thing that we need to bear in mind with the X chromosome, and this has to do with gene dosage. So this doesn't necessarily affect all chromosomes, but in the case of the X chromosome, it does. So for a female, having 2 active X chromosomes means that the gene dosage, how many active copies of the genes there are going in the cell, is too high. And so the way for this to be dealt with is that very early on in the development of a female mammal, which includes humans, one of the 2 X chromosomes in each nucleus of the cells is disabled. It's an extreme version of the condensation of the chromosomes that we saw back when we were talking about mitosis and meiosis. But it only affects one of the 2 X chromosomes, and it is condensed down into a very, very small structure that is sometimes referred to as a bar body. And it's actually visible inside the nucleus. You can actually see it in the nucleus because it's so tightly bound up. And every cell that is derived from these cells where this occurs has the same X chromosome disabled. This occurs very early in the development of the female embryos, usually around the 8 to 16 cell stage. And what that means is that when you look at the adult female, she actually represents a patchwork of X chromosome expression, or a mosaic as the book uses. Because if the female is homozygous for all of the genes on the X chromosome, it doesn't matter which X chromosome gets disabled. But if she's heterozygous, then it really does matter which of the two X chromosomes get disabled, particularly if those two X chromosomes lead to phenotypes that are visible. Now, in humans, it's really difficult to pinpoint a condition that might be associated with this what is known as X inactivation formation of the Barr body. But in cats, there is a condition that we know is specifically associated with this, and this is the tortoiseshell cat. So here you can see a cat that shows this particular pattern. I think that, you know, yeah, this is, I'm trying to remember if this is tortoiseshell or, because they sometimes call it something else. And I'm at a loss to remember what that is right now. But it turns out that one of the genes that affects coat color in cats is located on the X chromosome. If I remember, I'll let you know. But I believe this is called tortoiseshell. If the female is homozygous for the orange allele, then it doesn't matter which of the two X chromosomes gets shut down, and she will be a ginger cat, tabby cat. If she is homozygous for black, it still doesn't matter which X chromosome gets shut down. She'll just be a black cat. But if she's heterozygous, then it does matter, because different cells with different X chromosomes inactivated will eventually lead to different color patterns on the coat. And that's where you see this patchwork of colors as a product of which X chromosome got shut down. And about 96% So about 96% of all tortoiseshell cats are female. So where do the 4% males come from? They are X, X, Y. They also have an extra copy of the X chromosome. And the same thing happens with them as happens in the female. One of the two X chromosomes gets shut down. So if you ever come across a male tortoiseshell cat, you can be very confident that he is XXY. How he got to be XXY is something that we'll talk about a little bit later in this chapter. But for now, just understand that male tortoiseshell cats have a gender chromosome combination of XXY. One of the two X chromosomes gets shut down, just as we see here in the female cats, and that leads to them being tortoiseshell as well, depending on which of the two X chromosomes gets shut down. Same basic idea. So, time for another quick break. Here are our two concept check questions. A white-eyed female fruit fly is mated with a red-eyed male. What genotypes and phenotypes would you expect in the offspring from this cross? And a couple who are normal have a son with color blindness. What is the probability that their second child is colorblind? What is the probability if the second child is a boy? What is it if it's a girl? See you on the other side of the break with the answers.